Okay, so picking up with those secondary lymph organs and structures, we have the tonsils. Most of you are aware of that. You have 3 sets of tonsils. They are not actual organs, so we refer to them as lymphoid nodules, and that is an important difference. So we have some slides of this up in lab if you want to look at them. I haven't yet used them on a practical, but we definitely use those sagittal heads. These are, of course, strategically located right here. They totally surround this tube in which you're going to, oh gosh, nice swollen tonsils, in which you're going to have primarily your encounters, right? They're going to be respiratory and oral. So nasal cavity, oral cavity. Right, so here's lingual tonsil, which is just behind the tongue, which you can't really see here. Here are these palatine tonsils, which are surrounding, this would be your soft palate, okay? And then you have these lovely pharyngeal tonsils, which are also known as the adenoids. And they really are guardians to the eustachian tube that drains the middle ear or helps to equalize pressure between the oral cavity and the middle ear cavity. So guardians, if you will, of big collection of lymphocytes in here, but do not have that capsule that would qualify them as a true organ. And these are scattered throughout. So this is just your oral cavity. I said your main entry is going to be through that tube that brings food in. So the tube starting in the esophagus is lined, the inner lining that contacts food is called the mucosa. So you see this term mucosa associated. So with the mucosa, lymphoid tissue. And this is scattered throughout. The esophagus doesn't have any, because food just doesn't spend much time there, but food spent a long time in your small intestine. So we have these Peyer's patches that you can see on your models. The breasts, the lungs, the eyes even have this, because again, it's about contact and optimizing the ability to come in contact with the pathogen and therefore recognize it and start your response. Because I'm going to tell you right now, if a pathogen gets in unnoticed, every 10 to 20 minutes, they are doubling. And you're going to quickly be outnumbered by those pathogens if you don't find them. All right, so we're going to skip the goods for now and move into what we have as a barrier defense and the innate. So we're going to explain how this works, okay? So everything in this part is what we consider nonspecific. The barrier defense is nonspecific, right? The cells we talk about here, the soluble proteins, are nonspecific. Your only specific, again, is going to be those B and T lymphocytes and the antibodies that come from them. All right, so here I just remind you again, it's all about the ability to find and confront, right, a pathogen and prevent them from colonizing. So you versus the encounter colonization of pathogens in the body. Bacteria, viruses, complex microorganisms, these are all your what you're defending against. So the innate or nonspecific, right, general defenses do not change much, but they are there and ready to go, which is really important. All right, so they give you some of the examples of these guys right here in this cartoon, which is very nice, right? Surface defenses or barriers are skin, hair, and mucus, right? And then actual cells and proteins, we've got the mast cells and basophils, natural killer cells, complement system, that's proteins, phagocytes, right? So, and here's your adaptive, which is not, that's your third line, okay? And so adaptive immunity, so this is kind of a compare and contrast the 3 lines. T cells, B cells, right? So, adaptive or specific defenses are slower but more effective in the end. They have precise targeting. These guys, again, are the snipers. So the innate is going to assist the adaptive to increase the effectiveness. So you don't just say, okay, it's your turn, you guys, you take over this time, or you guys do all the work. It's a very cooperative effect. So again, I formalize it here. I said that a good defense requires that you identify or recognize, you contain the invader to the spot, right, and don't let it get all over the body. You kill it and you clean up, okay? So I say the idea, this is what you have to do to defend yourself, but really it's better just not to have to do that defense, which is why barriers are so important, all right? Keep it out, destroy it as it enters, flush it out. That's the motto of the barriers, right? Prevent this. So skin secretions, oral cavity, right? This is where we find these stomach, mucosal surfaces, and the normal bacteria you keep actually help defend you. So these are the specific entities, right? within these wares and how. So again, the skin has a whole lot of keratinized cells, and keratin is a hard substance that just keeps things out. And then cells of the Langerhans cells are also found in the epidermis, and they are actually going to be able to eat things, right? Skin secretions have their acids, and they just wash things away. In the oral cavity, we have lysozymes. In the stomach, we have acids. In the mucosal, again, you've got these cells that trap. Mucus is a trapping substance, so the mucus cells they're non-keratinized, but that substance mucus can trap stuff. And then there's competition from the other microbes in your body. You can't colonize a place if there's already people there, or rather microbes there. Okay. Cells of the innate immune response, again, so that's second line. So we said that second line, the innate, is it has phagocytes and it has soluble proteins. So here's our phagocytes. They're all part of this. They eat anything that's entered the body that they can find. All right. The most active are neutrophils, dendritic cells, and monocytes, which become macrophages. Macrophages move all around, are important in helping the adaptive response, but are not part of the actual adaptive. In other words, they're not the adaptive response themselves, but they are very good at assisting in cleaning up and eating things and killing. They are found in lungs, spleen, liver, bone marrow, connective tissue, lymph nodes, and lymphatic nodules. Really, any of the places that we're going to find our specific response, we're going to find macrophages nearby. Neutrophils, which are the second most effective phagocyte we have, they also have granules that help enhance this response, right? So those granules are things like bleaches and defenses and oxidizing agents, peroxides. They will help enhance the response as well as eat, right? That's what we mean by engulf, eat the pathogen. All right, natural killer cells, again, part of the nonspecific, is really good against virally infected cells. So viruses get into your body and they move into cells because that's where they can create more virus particles. So these cells recognize the chemical signature of an infected cell. But again, they don't have antigen receptors and they are not specific. And once they recognize that there is an infected cell nearby, they begin secreting these cytolytic, so cell-destroying substances called perforins. Perforins perforate. Perforation is punching a hole in something. And then they have this other chemical called granzymes, which enters and triggers apoptosis. So the granzymes basically tell the infected cell to die. Right? Apoptosis is cell suicide. And natural killer cells, some of these granules also help increase inflammation, which brings in more cells to help defend. All right. So again, these are non-specific and do not involve an antigen receptor. What does it involve? Well, there's a pattern of molecules that is common to your non-human cells, bacteria, right, viruses and so forth. So a pattern of pathogen-specific molecules. Many of these are carbohydrates, a certain polysaccharide. And we find these in the bacterial cell walls, in the flagellar proteins that the bacteria have that our cells don't have, again, and our cells don't have cell walls. And so there's pattern recognition receptors on these cells that are nonspecific. But again, they're not recognizing an actual antigen. So it's not part of the specific. This is limited in its ability for recognition because, receptor needs to recognize all pathogens, and there's only so much space on the membrane. So there's only a limited variety of these receptors in these nonspecific cells. And again, you're limited by how much you can recognize. And you don't really know the specific strain of E. coli or the specific strain of Listeria. You have identified that there's something foreign in the body and you need to do something. So again, limited number of receptors that are active against many pathogens. So it's not very good. You're limited in what you're trying to find. There are lots of different pathogens, and this means you're compromising your ability to recognize. And I say the adaptive, on the other hand, has a large number of different receptors that identify only one specific pathogen, which is why we have to have a lot of different B and T cells. All right, So soluble mediators of the innate immune response include cytokines and chemokines. So cytokines are signaling molecules, right? So telling the body there's something up here, that allow short distance communication. So you're calling nearby cells, and they change what's going on in the receiving cell. So the cell that encounters a cytokine. So cytokines would ideally be released from a cell that's encountered or been affected by a pathogen, and the cytokine could call in other cells and causes that other cells to change some of their genetics or their gene expression, which then makes them more effective at defense. Chemokines are similar, but they attract cells from farther away. So it's just really what's the distance of effect. Early induced proteins made as they are needed, and this is just another group of these soluble mediators. Interferons produced by infected cells cause the uninfected cells to make antiviral proteins. So interferons actually do interfere with viral replication. And so they're effective, but again, they don't know. They're just released from an infected cell, and this is going to tell the other cells nearby that there's a problem. And the nearby cells will then start to make their own chemicals, right, that will help to limit the infection. And so interferons are a type of cytokine. Mannose binding, well, mannose is this carbohydrate, and C-reactive proteins bind to these polysaccharides. Again, and this is commonly what you find in the wall of a bacteria. The cell wall of a bacteria are different kinds of polysaccharides. And so we're trying to go after we're trying to limit who we're going after, but we're not doing a great job here. But it's still effective, right? So they help bind bacterial cells for phagocytosis, so it gets them all together. But, it's easy to make a mistake with that because lots of bacteria have mannose in their cell wall. All right. So here is a specific group of soluble proteins called the complement system, right? And these are in your plasma right now as you are listening to this or reading this in the book, all right? And as well as some of the other fluids in your body. So complement, once it's activated, is irreversible. So again, what is happening now is your complement is circulating inactive, all right? And it has a very specific pathway of activation, which you will not have to know from me, but this should bring to mind a way that we get coagulation, a very specific sequence of activation of proteins, which then end up with causing fibrin to form. Well, in this case, they're going to end up with generally poking holes in the invading cell's membrane. So here's our invading pathogen, all right? Now, you'll notice here in this that an antibody really enhances this whole complement, and this is important for the activation. So complement is probably not going to be active until we get some of that cooperation with the specific immune response and the formation of antibodies. But once antibodies start binding to this antigen and we really know the specific invader, then complement becomes activated in a sequence, and it attracts in other eaters, and it helps to punch holes in the membrane of the pathogen to destroy it as well. So it's a very effective defense once it's activated. So binds to membrane of pathogen, marking it for phagocytosis, right? This is the mark, this is the target. Acts as chemotactic, so chemical collar to attract, and forms these pores in the plasma membrane. So complement is still nonspecific because the complement itself did not bind to the antigen. All right? The inflammatory response is another component of the nonspecific response. And in this case, again, inflammation occurs in any tissue injury. Inflammation always results in redness, swelling, heat, and pain. All right? And so what happens is the injured cells become the callers. They start releasing cytokines and chemokines. All right? And this results in, again, chemotaxis, in which neutrophils are the first to arrive because they are the most plentiful, and they will leave. So we're going from here to here. Again, neutrophils leaving circulation and coming in and consuming these rod-like pathogens, bacillus, right? Inflammation is going to not only contain the invader, it's going to clean up, and it's going to repair, because fibroblasts in the connective tissue around here are going to, right, bind up and stop this.